I'm Professor Philip Rudland. I'm a biochemist and a pathologist. I lead a team interested in studying metastasis in breast cancer. When a patient presents at a hospital with a primary cancer, the first step is for the surgeon to remove that tumour. If there has been no spread of that tumour to other organs, then the patient can go home and feel cured. However, if the tumour has spread, or metastasized as we call it, to other organs, then almost invariably, whatever the clinician does after that with treatment, the patient will die. Now, what we've discovered is a series of genes and proteins which can induce metastasis in experimental animal and model systems. These we call metastasis-inducing proteins. When our proteins are overexpressed in primary breast cancer, the patients die much more quickly of metastatic disease. These results have been corroborated by other groups and in other tumours. And our metastasis-inducing proteins now represent the most abundant class of proteins in metastases. We've been focusing on one particular, perhaps the most abundant one, called S100A4. It works by binding to the cellular cytoskeleton, causing the cells to migrate and invade. We have, with the chemistry department, synthesized chemicals to block these processes and armed this component or chemical with a warhead that destroys its target protein, in this case S100A4. This makes the chemicals much more efficient, increasing their efficiency over 10,000-fold. We now have a lead compound which is active at nanomolar concentrations in our cell and animal experiments. What we need to do in the future is to conduct a preclinical trial in animals and a clinical trial in humans. Hopefully, if the experiments are successful, we will have generated the first targeted anti-metastatic therapy.